Hello, everyone, and welcome. My name is Amna, and it's a pleasure to have you here today. Medicine is often seen as extraordinary, how a single pill can move through the body, target the right place, and bring about healing. While it may appear almost magical, the reality is that it is grounded in science. Today, we will uncover the principles that make this possible. Our focus is on an essential concept known as pharmacodynamics. Though the term may sound technical, at its core it describes something very clear: the effects a drug produces within the body. Together, we will explore this foundational idea. At the very heart of this entire story is something called a receptor. I want you to think of a receptor as a tiny, highly specific lock on the surface of or inside our cells. And the drug is the key. These receptors are absolutely central to how medicines work. First, they determine the relationship between how much drug you take and how much effect you get. The better the key fits the lock, what we call its affinity, the less drug you will need. And the total number of locks you have can set a limit on the maximum possible effect. Second, these receptors are what make drugs selective. Your body has countless different types of locks on different cells. A heart medication is a key that's shaped specifically for the locks on heart cells, not brain cells. This incredible specificity is what allows us to target certain parts of the body. And finally, receptors are the mediators of the drug's action. They are the targets for drugs that turn things on and for drugs that shut things down. Most of these receptors are proteins, and they come in a few different flavors, like enzymes or little pumps that move things across the cell membrane. So we have our drug, the key, and our receptor, the lock. What happens when they meet? This brings us to the dose-response relationship. It's actually much simpler than it sounds. If we were to plot a graph showing the drug concentration on one axis and the effect it produces on the other, we'd get a beautiful S-shaped curve. This curve tells us two incredibly important things. First is the Emax, or the maximal efficacy. This is the ceiling of the graph, the absolute greatest effect the drug is capable of producing. It's the top of the S. This is so important in a clinical setting because it tells us the maximum therapeutic benefit we can get from a particular medicine. The second thing is called the EC50. This is the concentration of the drug that gives you exactly half of that maximal effect. It's the halfway point up the S curve. The EC50 is our measure of a drug's potency. A drug with a very low EC50 is very potent because it takes only a tiny amount to get a significant effect. Now, sometimes a cell is so sensitive that you don't even need to occupy all of the available receptors, all the locks, to get the full maximal effect. When this happens, we say there are spare receptors. Think of it like a stadium. You might be able to get a full stadium roar with only half the seats filled. The presence of these spare receptors makes the system incredibly sensitive to the drug. Let's talk a little more about the different kinds of keys we can have. In pharmacology, we mainly talk about three types. First, we have the agonist. An agonist is the perfect key. It binds to the receptor, turns the lock, and activates it, causing a biological response. It's the go signal. Next, we have the antagonist. An antagonist is a key that fits perfectly into the lock but is broken, so it can't turn it. It just sits there, blocking the lock. Its effect comes from preventing the real key, the agonist, from getting in and doing its job. There are two main kinds of antagonists. A competitive antagonist is like playing musical chairs. It competes for the same spot as the agonist. If you add more and more of the agonist, it can eventually win the chair and overcome the block. So a competitive antagonist makes the agonist look less potent, but it doesn't change the maximal effect you can get. A non-competitive antagonist is different. Think of this one as putting super glue in the lock. It binds irreversibly and takes that receptor out of commission. It doesn't matter how much agonist you add, that lock is permanently blocked. This means a non-competitive antagonist will lower the maximum possible effect. Finally, we have a fascinating one called a partial agonist. Imagine a dimmer switch instead of a simple on-off switch. A partial agonist can bind to and activate a receptor, but it can only turn the lights up to, say, 50%. It can never produce the full 100% response of a full agonist. And here's the really interesting part. If a full agonist is also present, the partial agonist actually gets in the way, competing for the receptors and acting like an antagonist. Our cells have developed a handful of truly elegant ways to get a message from the outside to the inside. Let's look at the major receptor families. Some drugs, like steroids, are so tiny and lipid soluble they can pass right through the cell membrane. They find their receptor waiting for them inside the cell. Together, this drug-receptor pair travels to the cell's command center, the nucleus, and directly changes which genes are turned on or off. This process is powerful, but it's also quite slow. Another type is a receptor that is also an enzyme. When the drug binds to the outside part, it causes the inside parts of two receptors to come together and activate each other, starting a cascade of signals inside the cell. Receptors for insulin work this way. Then we have ligand-gated ion channels. These are essentially little pores or gates that open or close when a drug binds. This allows ions to rush into or out of the cell, changing its electrical state. This is the mechanism behind much of our nerve signaling, and it is incredibly fast. We are talking milliseconds. But the largest and perhaps most versatile family of all is the G Protein-Coupled Receptors, or GPCRs. When a drug binds to a GPCR on the outside of the cell, it activates a helper molecule on the inside called a G protein. This G protein then acts as a messenger, scurrying off to activate another protein, which then often produces something called a second messenger. These second messengers are tiny molecules like cyclic AMP that spread throughout the cell, amplifying the original signal 1,000-fold and activating a whole host of cellular machinery. It's an incredibly efficient way to turn a single binding event into a massive cellular response. And our cells are smart. If they are exposed to a signal for too long, they can become less sensitive to it. They might temporarily uncouple the receptor from its G protein, which we call desensitization. Or, if the signal persists for a very long time, the cell might actually pull the receptors inside and destroy them, a process called downregulation. It's the cell's way of saying, "Okay, I've heard the message." So how do we use all this incredible knowledge to help people? In clinical practice, we don't just look at how one person responds, we look at populations. This gives us what's called a quantal dose response curve, which looks at an all or none effect, like whether a patient's headache was cured or not. From this, we get two key values. The ED50 is the dose that is effective for 50% of the population, and the TD50 is the dose that causes a toxic effect in 50% of the population. The distance between these two doses gives us the therapeutic index. A large therapeutic index means there is a wide margin of safety between the effective dose and the toxic dose, which is exactly what we want to see in a safe medication. Of course, we know that people respond differently to drugs. This variation can happen for many reasons. It could be differences in how our bodies absorb or metabolize the drug. It could be due to genetic differences in our receptors themselves, or even because of changes in the complex signaling pathways that come after the receptor is activated. And finally, it's so important to remember that no drug is a perfect magic bullet. Drugs are selective, not specific. A drug will prefer one type of receptor, but it may bind to others, causing side effects. A side effect might be a simple extension of the drug's main job, or it might happen because the same receptor type exists in different tissues doing different things. Understanding this helps us design better, safer, and more effective medicines. So from the simple concept of a key fitting into a lock, we have this wonderfully complex and elegant system of signals, messengers, and responses that govern everything from our heartbeat to our thoughts. Isn't that just fascinating? We've learned that receptors determine a drug's effect, that agonists turn things on while antagonists block them, and that the dose we give is carefully chosen to maximize benefit while minimizing harm. I hope this has made the world of pharmacology feel a little more accessible and a little less mysterious. I would absolutely love to hear from you. What part of this was most interesting to you? Do you have any questions at all? Don't be shy. Let's chat about it in the comments below. I look forward to reading your thoughts. Until next time, stay curious. Bye for now.